what are the different terms that we come across in the genetics, and what is the meaning of this chapter, and etc etc. So chalo, so this is where we ended in the last class, correct guys? So we studied about what are dominant gene, what is recessive, what is hybrid, what is not pack cross and test cross. We have to see what it is today, and we will target this. Clear, no? So I hope the board is not too bright. Just give me a second. I'll try to fix this as well. Yeah. Chalo. So let's go back. So today we will try to stick to your NCRT, and then we will finish as much as possible with the given concepts in your textbook. And then what we will do, as I told you already, I will try to target some of the non-Mendelian concepts as well, as like with respect to which is not there in your NCRT and which will help us in some of the competitive questions. Okay, so first let's go with the genetics itself. So to understand genetics, what we have to do is we have to make sure that we follow the experiments that is conducted by the Mendel, and then we see how the results are drawn from this particular experimentation. Two things are very, very necessary for us to observe here. What is that? The first scenario that we have to consider is how well organized was Mendelian experiments. And the second part is how well organized this data was. Okay. So whatever he did, he maintained a very, very clean data throughout the whole experimentation process for the 7 years term. So now we know that Mendel popularly worked on the garden pea plant, right? So why did he choose the pea plant? Is the first question. The pea plant he worked was around 30,000 in numbers. Okay, roughly he worked with 30,000 plants. That is the first thing. So as I told you, whether it is a luck factor or whether it is a well thought process, we really can't guess that. But he chose it. With his choice comes why the pea plant was chosen. So here are some of the reasons. One is purity, pure varieties are available. What do you mean by pure varieties? If I talk about tall, I can get only homozygous tall or homozygous dwarf or homozygous white or homozygous purple. You get my point? So pure characteristics can be drawn through this plant. That is first thing. So how did he make sure that they are of pure varieties? So we know that we have 7 contrasting characteristics that were selected by the Mendel, correct, right? So that is height of the plant, the flower position, the pod color, pod shape, seed color, seed shape, etc. So we have 7 characteristics. So for each character, there is an opposite character. For example, if I take height of the plant as a character, we have 2 varieties. What is that? One is a tall variety, other one is a dwarf variety. So what Mendel did was, Mendel had the gametes separated for all the characteristics. That means he had very pure pollen grains having only capital T, pure pollen grains having only small t. Similarly, for all the characteristics, okay, he had very pure alleles segregated among them. So one thing is we can go for the pure varieties in the pea plant, first thing. Second thing is pea plants are easy to cultivate. What is the easiness of cultivation? One thing is its lifespan has to be very, very short. If the lifespan of an organism is over a decade, okay, let's say it's like 10 years or 5 years or something like that, what would happen? How would it affect the experimentation? See, if I choose an organism with its short lifespan, gives its capacity to reproduce several times within that very short period of time. If I take the pea plant as an example, its lifespan is around 3 months. So within these 3 months, I can have many cycles of reproduction. So I can make sure that within a short period of time, I get my results. I get to see my results as quickly as possible. If I take any other animal which lives for many years, then it has to grow through the juvenility, it has to come through the reproductive phase, and then the reproduction process also would be complicated. So that's why the easiness to cultivate the pea plant was very, very very, very feasible or very, very acceptable compared to that of other options. Okay. Next one is, as I told you, it's only few months, like around 3 months for the whole life cycle to complete, so we can get the results very easily. Next one is contrasting traits are observed. This is a very, very important point. As we explained, as yesterday I explained you, what is the meaning of contrasting? Contrasting means you have the oppositeness. There is no middle character to it. Correct, right? So now you understand. There is only tall or there is only dwarf. There is no medium size. You understand my point. So these characteristics were very well defined in pea plant. So that's why it was very easy to analyze the data. Next one is this one. Flowers are bisexual. So what is bisexual? So they have both the male and female parts in the same flower. So that is why the self-pollination has to be, they get self-pollinated. For the cross-pollination to occur, they have to be done manually. So what is the significance of this? So that means the pollen pollution, what we call it as in genetics, what will happen? We have to make sure that our whole experimentation setup is not disturbed. Correct, right? Why it is not disturbed? Imagine you have so many plants grown in a garden, okay? And you know that it's an open nature. It's vulnerable to the bees, it's vulnerable to the ants, it can be pollinated. So they're all pollinators, right? So you cannot make sure that 24 hours you have the keen eye to make sure that they are not contaminated by these pollinators, and they bring any some random pollen grains, and then they ruin your experimentation. So this headache was completely avoided by the nature of the plant itself. So the nature of the plant is it is bisexual plant. So usually the best possibility that can happen is the same characteristics will get transferred into the same plant. So what will happen in self pollination? You know that you have a flower and you have the a stigma. Okay, and then you have your anther. So the pollen grains can go from anther to the stigma of the same plant. That means the characteristics get circled around rather than the new characteristics being introduced accidentally. Correct, right? So that is why this particular, that is flowers are bisexual and normally self-pollinated, is a very important and very good characteristics with respect to the pea plant. Next one is flowers can be cross-pollinated only manually. So this is another important key feature why it is a very beautiful plant. Next one is hybrids are fertile. So every single point makes it such deep importance and such deep significance. See what will happen. You can see that in market, whenever we find some of the hybrid plants for the cultivation, you can only grow them for once. Only one life cycle it will be given. Why? Because some plants are designed like that by the companies, or some plants nature itself is like that. So when I say that some plants are nature is like to be fertile in a hybrid situation. So what will happen whenever you mix two different characteristics, which is non-natural in occurrence? So in naturally it does not support that system. So it does not go for the dwarf to tall, tall to dwarf. It does not go because they are self-pollinating plants, right? So when they are self-pollinating means they only accept their own pollen grains. So at this situation, what will happen is the possibility of the other pollen grains coming and fertilizing them is quite rare. So when these rare things happen in our plants, the plants usually in the next generation, they won't show the fertility. Fertility means they won't produce healthy pollen grains that has the capacity to reproduce. So this is very common in nature. But very special characteristics of pea plant is even the hybrids are fertile. When the hybrids are fertile, that's when you can go from F1 to F2. You understand my point? So that is why the pea plant was such a very beautiful plant for experimentation in genetics. So yesterday we discussed about what are the characteristics that he chose for the experimentation. So 7 contrasting characteristics: seed shape, seed color, pod shape, pod color, flower position, and plant size. And these are the contrasting characteristics with respect to them. Okay. So let's go with the experimentation itself. Now, the first thing what Mendel does is he selects one character. So first we go with monohybrid cross. In monohybrid, what is monohybrid cross? A single, okay? Mono means, for example, inheritance of one gene also is another term for monohybrid cross. Mono means it is single, correct, no? Hybrid means mixing, single mixing, correct, no? So what are we mixing? Single, what single mixing are we doing? A character. So what is a character that Mendel is choosing? See, we are taking an example of a character. So our example to understand is the height of the plant. You understand? That doesn't mean that Mendel concluded his monohybrid cross only with the height of the plant. He might have worked with other characteristics as well, such as flower position, flower color, seed shape, seed color. All of these characteristics he might have worked with. But for our understanding, we are taking one character, okay, among them. What is the one character that we are trying to look at it? It is the height of the plant. So if I see the height of the plant, correct? No, height is a character. So in this height, I have two possibilities. What is that? One is a tall plant, other one is a dwarf plant. So I take one character and I mix these plants, that is one tall plant and one dwarf plant. So in a monohybrid cross, what happens is, okay, one second. In a monohybrid cross, first I go with the parental generation. In a parental generation, what I have to do is I have to choose a male plant and I have to choose a female plant. Anyways, either you can tell it as male plant or female plant, okay? Or you can call it as gametes also, it's fine, okay? So what will happen when I choose my parents, they have to be of homozygous nature. So why homozygous nature? The purity of alleles should be maintained so that I take the pure forms and then I mix it so that I see the observations in the hybrid form. So what is the monohybrid varieties here? Sorry, homozygous varieties. For tall plant, okay, my alleles are homozygous. That means both the alleles are capital T, capital T. And then for dwarf nature, I choose my alleles which are again homozygous, that is small t and small t. Understood? So my parents are in diploid form. My parents are in what? Diploid form. That is why when I said diploid form, you should understand this by now. Yesterday I explained why we take alleles as pairs, not as single. Correct? No. Now I take the parents and the parents are represented as capital T and small t, small t. Now, in the parent's DNA or in the parent's chromosome or in the parent's genetic makeup, when you see, what you can observe that both the parents are having same type of alleles. That is either capital T in one parent or small t and small t in another parent. Now let us assume that our capital T is a male plant and then our small t is a female plant. Okay, we are assuming that this is a male plant and this is the female plant. What would happen if they produce gametes? If my male plant starts producing pollen grains, okay, no? Sorry, if my male plant starts producing pollen grains, what are pollen grains? Pollen grains are gametes. So are gametes haploid or diploid? They are haploid in nature, correct, right? They are haploid in nature. So if they are haploid, what would be the chromosome number, N or 2N? Tell me guys, what would be the gamete number? Is it N or 2N? What do you think? Is a N? Exactly. So if they are N, so the 2N nature, this is 2N. In their body, they are 2N, correct, right? So when they produce the gametes, what would be the number of chromosomes? N. So they would produce the pollen grain, which is of N number of, chromosomes. So now tell me, out of 100% of the pollen grains produced, would all the pollen grains have the same type of alleles? Yes, right? So that is why we choose only T. Okay, so that means all the pollen grains will have the chromosome containing T type of an allele. Okay, so you understood. Now they will TT and then they produce pollen grains. The pollen grains are haploid. That's why we choose not double, we choose one. Why we choose one? Because there is no other possibility that these pollen grains would have another type of an allele. As simple as that. Now the same way goes with the dwarf plant. The dwarf plant, excuse me one second. Yeah. Is it too bright? Students, are you able to see the screen properly? Are you able to see the words? Please confirm. Fast. Is the screen clear, guys? Yes? Okay, okay. Okay, thank you. And I'm sorry for the disturbance. Yes, yes. Okay. So now the same way goes with the dwarf plant, guys. Okay. So the dwarf plant, the plant is diploid. The dwarf plant is a female, and she produces what? She produces eggs. The eggs are haploid, so they are n in number. So all the alleles, okay, in the egg would be of similar type of allele. That is our small t. Simple as that. So now we understood. Now we understood that how we are taking either NN, so that is capital T, or small t, how it became a single T and a single small t and a single capital T. So let's get back here to the cross. In a monohybrid cross, we understood the nature of how the alleles are selected or how the alleles get segregated. Let's see here. The parental generation, we take capital T, that is a tall plant, and we take a dwarf plant. They are homozygous tall and a homozygous dwarf, and they produce gametes. When they produce gametes, 100% of the gametes are either capital T or 100% of the gametes are small t. So when they produce offsprings, the first generation of the offsprings are called as filial 1 generation or F1 generation. Okay, we call it as filial. They're called as filial 1 generation. They're called as children. Okay, the children is called as F1 generation. So F1 generation, what will happen? Now we have capital T was the parents, and they were crossed with small t and small t. Now they produce 100% capital T and then 100% small t as a type of gametes. Now when they cross, the only possibility is one pollen grain with a capital T will mate with another egg with the small t. So all of them will have the genotype as capital T and small t. So this will be our F1 generation. So now, after getting the F1 generation, all of them were of capital T and small t type. Now, phenotypically, when Mendel saw the results, he saw that all the plants were tall, and this confused him a lot. So he was worried. Why he was worried? Where did the dwarfness go? So what happened to the dwarf character? So he was quite amazed by this. One of the character went missing like that. So he wanted to investigate. So through his investigation, what he did was he takes the F1 plants. What is our F1 plants? Let's assume that we have an F1 plant, and F1 plant's genotype is of what type, guys? Capital T and small t. Now we will understand even more clear. If my F1 plants produce gametes, what would be the possibility? The possibility is there is a possibility that 50% of my gametes would be of capital T type and 50% of the gametes would be of small t type. Do you agree? If a plant having a genotype of capital T, small t produce gametes, what would be the possibility? They might produce out of 100% of the pollen grains, 50% might have capital T and 50% might have small t. As simple as that. Now what he will do? He will self-pollinate. What is self-pollination? We transfer the pollen grains from the anther of a same plant to the stigma of the same plant. That is called as self-pollination. Correct, right? So when I self-pollinate, what will happen? My capital T, small t would be crossed with capital T and small t. Correct, right? So now, sorry. Now what is the possibility, guys? Now in the capital T, small t plant, excuse me. Yeah, in the capital T, small t, here I have 50% as capital T, 50% as small t. Then I will cross with 50% of capital T eggs, 50% of small t eggs. Are you following me? How do we collect gametes? Now what will happen? If I cross them, okay, there is a chance that this capital T would meet this capital T egg. This capital T pollen grains would meet the small t egg. The small t pollen would meet the capital T egg, and the small t pollen would meet the small t egg. Correct, no? If I consider this as pollen, and this, if I consider this as egg, or male and female, you can take it for your convenience. So what are the possibilities, guys? After the cross of the F1, if I self cross the F1, I will get capital T, capital T, capital T, small t, capital T, small t, and then I get the small t and small t. So finally, what I see is out of the F2 generation, the children, the grandchildren, that is the F2 generation, what I find is I finally see a dwarf plant coming into existence. In the F1 generation, the dwarf character was completely overshadowed. It was not visible at all. But in the F2 generation, what I see is there is the visibility of the dwarf character. So what Mendel did, Mendel started understanding this in a statistical sense. So how would he analyze the data? If I see the physical characteristics of an F2 generation, what I see is 3 of the plants that are born are of tall nature, correct? And one of the plant that was born is of dwarf nature. That means out of the 4 plants, 3 plants are tall, 1 plant is dwarf. So they have a characteristic. So there is a ratio for it. What is the ratio? Out of the population of 4, 3 are tall, 1 is dwarf. So which we call it as a phenotypic ratio. The phenotypic ratio of the monohybrid cross in the F2 generation is how much? It is 3:1. Correct, right? This is the phenotypic ratio. 3:1 is the phenotypic ratio. Whereas when it comes to genotypic ratio, what would be the genotypic ratio? The genotypic ratio would be, excuse me, what would be the genotypic ratio? 1:2:1. So, easy to tell the ratios, but to analyze the ratio would give us more idea. So, the genotypic ratio is what? 1:2:1. Correct, no? So what is 1? 1 is capital T, capital T. What is 2? Capital T, small t. And what is another one? Small t and small t. So this is how the plant's genotype would be. Now, Mendel got this ratio, genotypic ratio and phenotypic ratio. What is the results drawn out of the monohybrid cross? So we understood monohybrid cross. Now, when he conducted the cross, when Mendel, imagine that Mendel conducted the cross. When he conducted the cross, he has no idea that alleles have to be taken as pairs. He had no idea. He could have taken as capital T, capital T, capital T, right? He would have taken it as triplets. Or he would have taken it as quadruplets. Or capital T, capital T, capital T, capital T, 4 he would have taken. Or he might have taken 8. Or he might have taken single. Why he went for only 2? Now look at in the possible math sense. In the math sense, the only way you can predict this ratio would be if you take it as pairs, correct, right? Only if you take the factors as pairs, your ratio would end up as 1:2:1, correct, no? Or even if you take it as multiples of 2, like 2, 4, 6, 8. Finally, when you divide the ratio, it would again end up as 1:2:1. So the simple way, simple mathematical sense he used, if I get the ratio as 1:2:1, then the alleles have to be what? They have to be present in pairs. So that's when he comes up with the idea of law of paired factors. So what he says, what is the understanding of paired factors? If I see an organism, if I take any organism, if they have genes, any organism would have genes, right? If they have character, they have to have genes. What are genes? Genes are nothing but alleles. Correct, no? If I have a character, what is my character? We are talking about height. So if I have a character, then I would be having an allele. So if I have an allele in a normal condition, in a diploid state, they should be present as what? They should be present as pairs. So that is what he says. So whenever we have factors which are responsible for transmission of characteristics, which any factor is there means they have to give rise to a character. So if we have a factor, they always have to be in what nature? They are present in paired forms. Okay, so they are always present in pairs. Got it? That is called as law of paired factors. So in a monohybrid cross, the first thing he understood was if there are alleles, they have to be present in pairs. Very important concept. Okay, next one is law of dominance. Now see here, even though law of paired factors and law of dominance, they are not real laws of monohybrid cross. We are not still talking about a law. They are a condition. We call it as condition of dominance, condition of paired factors. It's not law yet. Understand this point. Okay. So what is law of dominance? Law of dominance states that whenever we have two alleles of dissimilar nature is also called as what? Heterozygous. Correct, no? Heterozygous. What is heterozygous? They are of dissimilar. They are not similar. So what is the opposite of heterozygous? Homozygous. We have capital T and small t. In this condition, we cannot analyze anything. In a heterozygous condition, we can come to a conclusion. What happened in F1 generation? Alleles were in heterozygous condition. So that is, there was capital T and small t allele living together in a same organism. So, or being together in a same organism. When they are together, when they are in a heterozygous condition, only one allele expressed over the other allele completely. The other allele's presence was not at all recognized. We never could be able to understand if there was small t. Correct? No? Would you understand that there is small t? No, because the tallness is very similar to that of a parent. So it was completely overshadowed, completely dominant. So how would you tell that in the dominance case? In the dominance condition, in a hybrid condition, or in a heterozygous condition, when two alleles are present, one allele expresses completely over the other allele. That is called as law of dominance. Got it? No? So this is called as law of dominance. Next comes Mendel's real first law. What is the real first law? A law of segregation. So this is the first law of Mendelian genetics. So if anybody asks what is the first law, you should tell that it is law of segregation. Law of segregation is the proper law, okay, proper Mendelian first law of inheritance. So according to this, what does it say? It says that whenever alleles are in heterozygous condition, so we took that capital T, small t, correct? And then we crossed with capital T and small t. When we did that, what happened? Even though in a heterozygous condition, we had dissimilar alleles, correct? No, we have a capital T and small t and a capital T and small t. Both are dissimilar, correct, right? They were in a heterozygous condition. But during gametogenesis, when they produced gametes, we had what did we have? We had capital T type of gametes. We had small t type of gametes. Let's say that this is male gametes, correct? And we also could be we also got capital T type of gametes and small t type of gametes even in eggs, correct? No, in male also we got both the type of capital T as well as small t. In female also we get as capital T and small t. So what does it explain? Whenever alleles are in heterozygous condition, during gametogenesis, okay, they segregate independently of each other. It doesn't matter whether they are in the very dominant allele or very recessive allele, it doesn't matter. But when the gametogenesis happen, they segregate independently such that, what did we get? We got a dwarf plant here, correct? No, we got a dwarf plant here. The dwarf plant, the existence of the dwarf plant was only possible if they segregated independently, correct? No. So this is what is law of segregation or law of purity of gametes. What does it say? Whenever alleles are in heterozygous condition, they segregate independently of each other. It doesn't matter whether it is dominant or whether it is subdominant or codominant, it doesn't matter. So it keeps on segregating independently. So this is called as law of segregation. I know it is quite slow that I'm teaching you this. If you understand how we analyze pairs, how we analyze characteristics, how we analyze gametogenesis, next all of them would be easy. Now, did you understand till now? Please, can you confirm? Can you please confirm, guys? If you have understood till now, next would be faster. Okay, tell me. Okay, understood. Thank you. Others, if you have doubt, please feel free to drop your doubts. And I have a very good strength today, and I'm very, very happy that you guys are all present here. Okay. Remember, you are in a Enable Plus batch, and it is our responsibility that we build a very, very good relationship. I know who you are, and you should know who I am throughout this year and even in the retention time, so that when you meet me physically in the retention, okay, I should not be meeting strangers. I should be knowing what is my caliber of my student. I should understand, okay, this student is this much, he has doubts here, he has problems here, so we can sort day by day, and I should make you improve each day. That is only possible if you build a proper communication, guys. So make sure that you feel that you're in a real class, you feel that, okay, I can approach this guy and I can ask my doubts, and I make sure that he knows me and I know you. Okay, got it? Chalo. Now the next concept is the back cross. Mendel's genius is again appreciated here. Now we understood. See, guys, just imagine this experimentation. In this experimentation, if Mendel was not able to analyze the purity of gametes, when I say analyze the purity of gametes, if he did not understand the difference between capital T and small t, what would have happened to the whole experimentation setup? Would he be able to understand the difference? Here also we get a tall plant, correct? Phenotypically, here also we get a tall plant. Here also phenotypically we get a tall plant. How was Mendel able to differentiate that this is a monozygous tall plant and this is a heterozygous tall plant? That is another smartness of Mendel's genetical experimentation. What he did was he understood the significance of backcross. Okay. So whenever I talk about a backcross, okay, in a backcross, I can get two types of cross. Okay, this is a backcross. So what is a backcross? Before we understand a test cross, we talk about a backcross. If I have a parent, okay, I have a parent, and I take a parent and I cross with a B parent, and I get a C. What is a C? C is my F1 generation. C is my F1 generation. That is a children that of the A and B. That is a C. If I take a C plant and I cross with either A or either B, this is called as a back cross. It doesn't matter if I cross with the A plant or if I cross with the B plant, that is called as what? It is called as a back cross. You understand? I take an F1 generation, I take a children and I cross it with a parent, irrespective of what nature is of the parent. Okay, you understand when I say what nature is of the parent? It can be a tall plant, it can be a dwarf plant. I don't mind. I just cross it. This is called as a back cross. In a back cross, you cannot draw any significant results. Okay, you can only when you differentiate a back cross. A back cross can be divided again into two different types of a cross. One is an out cross, other one is a test cross. What is an out cross and what is a test cross? You know what is a test cross, but first we talk about what is an out cross. Correct? So what is an out cross? Let's say that we have a capital T that is crossed with a capital small t and a small t, correct, right? That's what we did. That's what we did. Now, what will happen if I take the F1 generation, which is a capital T and small t, now I cross with a homozygous dominant plant. If I cross with a homozygous dominant plant, what would be my result? My result would be capital T, capital T, capital T, capital T, capital T, small t, capital T, small t. Do you guys agree? Do you guys agree with the results? So what happened here? I just took my F1 generation and I crossed with the dominant plant, homozygous dominant plant. So here, this cross is called as an outcross. In this outcross, for me, I would not again be able to understand what is the genotype of my F1 generation. Will I be able to analyze? All of them are tall plants. All of them are tall plants. Whether this F2 generation that I've got, I would never be able to understand if, okay, if they have a homozygous character or if they have a heterozygous character. If I give you 4 tall plants here, by looking at them, would you be able to tell what kind of a genetics they have? It's impossible, isn't it? So that is what is called as an outcross. Now, what is a test cross? Test cross is when you specifically choose an F1 plant and then cross with the homozygous recessive plant. So that is where the test cross significance comes. So what is the use of a test cross? The use of a test cross gives us an ability to understand the genotype of the F1 generation. Now look here, in the parental generation, what did we take? We take capital T, and then we take the small t and small t. In the F1 generation, what happens? We take the capital T and small t. That is the result of the F1 generation. Now we choose this child from the F1 generation and we cross it with small t and small t. What is our small t? It is a homozygous recessive. Okay? Homozygous recessive, very, very important. We specifically choose a homozygous recessive plant. Correct, bro? So when I take the F1 generation and I cross with the homozygous recessive only, then what do I get? What is my cross, student? What is my result? The result is that I take a capital T and a small t and I cross it with a small t and a small t. I will get capital T small t, correct? Capital T small t, and then I get small t and a small t and small t. Now what happens? If you compare the results of an outcross and a test cross, in the outcross, the F2 generation is all tall plants. In a test cross, it is 50-50. You get two of them are tall plants and two of them are dwarf plants. So the dwarf nature again comes into light. So if a dwarf plant again comes into light, that means you have conducted a test cross. Okay. Now, one more question we take. What happens? What happens? Okay. If my plant, let's say, accidentally, I make a mistake. And the mistake I conducted is I took a tall plant, random tall plant, and that plant is of homozygous nature. Now you know the results. What would be the F2? All of them would be tall. Okay. Only if my F1 generation is of capital T and small t, only then I would get a 2:2 ratio. That is, two of them are tall, two of them are dwarf. Then I would have conducted successful test cross, and through test cross, I can understand the purity of alleles. Okay, so whether my alleles are in a homozygous nature or whether my alleles are in a heterozygous nature. So test cross is a brilliant way in which Mendel was able to differentiate if his plants were in the homozygous condition or if his plants were in heterozygous condition. And through homozygous condition, he was able to extract pure pollen grains. Okay, in the beginning I told you he was able to segregate all 14 different types of pollen grains separately. He boxed them. Okay, this is tall pollen grains, this is dwarf pollen grains, this is yellow, this is wrinkled. All of them he had separate pollen grains. How was he able to do it? He conducted meticulous test cross for every single character and he segregated them very properly. Okay, so that is what is test cross. Okay, got it? No? Chalo. Now, some of the cases, what happens is our Mendelian laws or Mendelian genetics will not be applicable. Okay, it's not followable. Why? Because the randomness in the nature is too wild. I can tell the colors in your color box, like a painting. You cannot expect the black and white kind of a nature in your surrounding. The nature is always comes with surprises. It's always about differences. Correct, no people? So that's when we have some of the ways, some of the conditions that we can observe where the Mendelian genetics cannot be applicable. Okay, one of the examples is incomplete dominance. So if I take a monohybrid cross, in a monohybrid cross, I can clearly come with law of dominance. Correct, no? I can clearly come up with the idea of law of dominance. But in an incomplete dominance, the law of dominance is not applicable. We are taking the same character. What is the same character? Sorry, one character. What is one character? In a Mendelian first cross, we took the height of a plant as one character. Let's take another plant. The another plant that we are taking is a snapdragon or a dog flower. Now, in a dog flower, what will happen? We are taking as a color of a flower as an example. So in the color of flower, we have two flowers. See, we have a red flower, we have a white flower, and then finally we are getting a purple flower here. Okay, and then here we are having a red flower and we have a white flower. Here we're getting a pink flower. Okay, so these are the two examples. See, this is a Mirabilis jalapa, that is a 4 o'clock plant. This is how the flowers look like. And here you see, this is an example of the dog flower where we have two different plants and this is a different color we are getting. So let's analyze the results. Okay, so what happened here? The parental plants were very simple. We take a red plant, red flower plant, and we take a white flower plant. Let's say that the red is a dominant. We cannot tell it is a dominant, okay? Because we don't know the results yet. So what is happening? Let's understand that we put red as capital R, and we take white as small r and small r for our convenience. And then this is the parental generation, no guys? Now we already know they produces what? Gametes. When they produce gametes, 100% of the R will give capital R. And then small r will give 100% small r. Correct? No. Now F1 generation, what will happen? In the F1 generation, I would get capital R, small r. Now here, I did not get red flowers. I got white flowers. In the F1 generation, all of them were pink flowers. There were no red, there was no white, but it was a mix of red and white. How beautiful is it? You can get any color, but you got pink. Just like how you mix the red color in your paint box and the white color in the paint box, it gets pink. Just like painting. Unbelievable. It can be of any pigmentation. Just like how we do the painting in our, if you do the drawing, if you paint, you understand that what I'm trying to tell. It could have been any color. Why pink? This is just like God is painting. He just used the two colors and then he mixed up and you get a pink color. Correct, no? So you get the pink color. Now, what happened here? Let's go for the F2 generation. If I self-pollinate, again, capital R, small r, and then I take capital R, small r, what would be the result? I will write it here, guys. Okay, capital R, capital R, capital R, small r, capital R, small r, and then what do I get? I get small r, small r. Now, this is red. This is pink. This is pink. This is red. This is white, sorry. Now, this is my genotype. This is my phenotype. Correct, no, guys? This is my genotype and this is my phenotype. Now, if you see the monohybrid cross, the phenotypic ratio was how much? 3:1. And what was the genotypic ratio? 1:2:1. Now look at the condition in monohybrid cross. This might be a question. In which of the monohybrid cross you have the same genotypic and the phenotypic ratio? Now this is a condition. You see, the phenotypic ratio is also 1:2:1 and the genotypic ratio also 1:2:1. How did we get this? See here, this is a genotype, agree. This is a genotype. Capital R and small r genotype is 1. Capital R, small r, capital R, small r, this genotype is 2. Small r, this genotype is 1. So 1:2:1. Agree? Again, you come to the phenotype. Phenotype means I can observe the color of the flowers and I can analyze. So 1 is red flower. 2 of them are pink flowers. 1 is white flower. Now this is the genotypic ratio. That is the phenotypic ratio. 1:2:1, 1:2:1. Now what happened here? So I did the same type of a cross that the Mendel did. Correct? No? This is also a monohybrid cross. This is also a cross with one character. The character I chose was a flower color. So flower color in a different plant, that is a snapdragon or 4 o'clock plant or dog plant or mirabilis jalapa. I take this and I conduct a cross. When I conducted a cross, I chose 2 parents. One is a white parent, another one is a red parent. When I crossed red flower with a white flower, I should have got red flower, but instead in the F1 generation, I end up getting a pink flower. And then I was not satisfied with the result. I go for the F2 cross, same way, self-pollinate them. When I self-pollinate them, I get capital R, capital R, capital R, small r, capital R, small r, and then I get small r, small r. Finally, if I observe the ratios, okay, if I observe the ratio are deviated. That you can see the deviation in the ratio. That means whenever there is a deviation in mathematics, you know that it is not logical. Correct? No, there is some logic missing. So that is where you understand that in an incomplete dominance, both the genotypic and phenotypic ratios are same. Whereas in the law of dominance, the genotypic and phenotypic ratios are very different. Why? Here we go. See what happens with respect to incomplete dominance. Incomplete dominance is also called as partial dominance, or it is also called as semi-dominance condition. Whenever you have the genetics or genes which are together, okay? So let's say we have an A gene and then we have a B gene together, okay? I think this is not so visible. I will go for this, okay? So whenever we have 2 genes together, there is a chances how the genes get expressed. So do you know how the genes function, guys? Yes or no? Is molecular basis of inheritance taught to you? Yes? Can you confirm? Is molecular basis of no? Yes. Okay. Good. Good. So is it? I think it's the same for everybody. Okay. So let's say now the question is, how do genes function? That is my question. Okay. Genes expressed by producing protein. Absolutely correct. Correct. No. So the expression of genes comes through protein. So a protein can be a hormone. For example, in height of the plant, we saw that there has to be a height hormone. So in capital K, in whenever there is a T, capital T, there is a height hormone produced. Wherever there is a small t, it does not have the capacity to produce height hormone. That's why the plant remained in a dwarf situation. So this is our case. Correct, right? So now what we analyze is how do genes express, especially in an incomplete dominant situation. So we know that if we take a condition of capital T and a small t condition, here you can see that there is one gene, that is capital T, which is producing 100% of the protein, whereas the small t does not have any role. It does not produce any protein at all. It does not produce any protein at all. That is why only when there is small t and small t you can see the dwarf nature, but as even if there is one capital T, you can see that there is a complete expression of height. So that means when one gene completely expresses, that means one gene produces the protein, other gene does not produce the protein at all. This is the one situation, guys. Second situation, we have a capital T, capital R and small r situation, whereas in the pink color plant, right, in the pink color, what is happening here? In this happening, you can say that capital R is also producing some protein, small r is also producing some protein. But what is happening is their combined effect is seen. Okay, their combined effect is in such a way that both of them are not independently expressing, they are combinedly expressing. Okay, so their expression is interdependent. You can see here, right? You cannot see the red spots and the white spots. If you see the red spot and the white spot, that means they are co-dominating. That means they are co-dominating. So R and R together are combining and producing one pigment. What is that one pigment? Pink pigment. You can consider that pink capital R is producing some 60% of protein, that is 60% of expression, and then you have the small r that is producing 40% of the expression. Any ratio you can take, but eventually what is happening, they produce one phenotype, mixing their product. The R, capital R product is also important, the small r product also important. They both combine together to give one product. What is that one product? Pink is the product. If they did not combine their expressions, we would have got red and white spotted plants. Agree with me, right? We would have got red and white spotted plants, but we never got a red and white spotted flowers. We got a pink flower. So what does it say? In a genetic expression, when the two alleles are expressing each other, the alleles produce a combined phenotypic expression. That is called as an incomplete dominance. We cannot understand who is dominating over who. You understand my point? Who is dominating? Can you tell me? If you put a red color more and a white color less, it would give dark pink. Correct, right? If I put white color more and a red color less, I would get light pink. Correct? No. Now, would you eliminate any of this one color? Pink is of significance. You don't want whether it is a dark pink or a light pink. So when you look at a flower color, you just go like these both are combining together to give us one effect. This is another expression. Let me give one more example. One more example is the blood group. You have the AB blood group. In AB blood group, you have AB blood group. Anybody here? Okay. Mine is O positive, guys. So others, if you have AB blood group, you know the blood groups, right? A, B, O, and AB. A blood group, B blood group, O blood group, and AB blood group. If I take AB blood group, now you see here, this is a codominant condition. What happens here? On the red blood cell, okay, on the red blood cells, on the plasma membrane, I have both types of the glycoproteins or the polysaccharides. What type of? Both A type of as well as B type antigens are present, correct? No, both A antigen as well as B antigen are present on the red blood cells plasma membrane. Now, do you see a combined effect or a separated effect? You see a separated expression, but they both are equally expressing 50-50. That's it. So this is called as a codominant condition. This is how the genes express, the possible ways in which the gene express. One completely takes over the other and gives only one effect, one phenotypic effect. Second one is you have two different genes acting together to give only one phenotype that is completely different from the parents. It is not tall, it is not dwarf, it can be medium. Similarly, it is not red, it is not white, it is pink. So this is called as a combined genetic effect to give one phenotype. Next one is AB. You take care of your business, I take care of my business, but phenotypically both the characteristics are expressed equally. That is the AB antigens present in a AB blood group. Understood, right? Did you understand how the genes work? Okay, our time is up, guys. Sorry, I had to take a little bit of time again in explaining few things. Crosses, right? So you have any doubts, guys? Did you understand today's class? Is it satisfied? Yes. Thank you. And as usual, please, we have too many people here, okay? And I would see what happens, as I told you before, if you don't give the feedback, that means that I have not, what do you tell? I have not taught the class at all. You understand? That means the class was not held at all. So that's why we don't go to that situation, right? So all this time we had good conversation. I hopefully, okay.